Yeah, so what I want to talk about now is how we use these QMMM simulations to do an application. It was to a very fundamental reaction that takes place in biology. It's one where you set the genetic code, so it's really challenging, both for even a classical mechanics simulation, because it involves protein and RNA complex. And before I even get started with my lecture, I think let me go to the credits. So I think you must know by now the reason we're able to do this workshop is that we worked for about a year on this paper that appeared in Nature Methods called NAMD Goes Quantum. And it really took a number of people to get it to work. And I think this workshop is very, very lucky in that at least half, or more than half, of the authors are in the workshop this time. So, of course, you've heard from Marcelo. You may have heard something already from Rafi. I'm not sure if you talked yet. But he will be explaining some of the free energy calculations and how difficult they are when you're using QMMM calculations. You heard from Till already. And, of course, this sort of NAMD going quantum was one of the last projects that I did with my husband, Carl Schulten, before he passed away. We thought, okay, I'll have Marcelo, who's a graduate student with me, and Rafi, who's a postdoc with my husband. We'll get together, we'll do this, it'll take 6 months, we're done. Ha-ha. Right? It took a lot more work. And then the other people that are involved in there, well, you also, a lot of them are connected with our NIH resource. So you need to work with the NAMD programmer. That was Jim Phillips there. How to get up these orbitals into VMD that takes interfacing with John Stone, who you may or may not have met. But we also had help from some students who were just getting, I want to do it a little bit more quickly. So they were even like sending scripts in to us to be able to get this done a little bit faster. And of course, we're really lucky. We had two of the leading quantum chemistry PIs in the world helping us. You've already met Gerd Rocha. So he was in charge of all the semi-empirical work. And then the fellow sitting, or the picture next to him, is Frank Dessa. He's at the Max Planck in Germany. And he is one of the developers, or his lab has developed ORCA. That's extremely, that we have these two extremes packages there is really a very important thing. And then, as I said, I think the reason we can also do this workshop is because of QuickMD. Listening to your questions, there's so many questions about how you set up these QMMM calculations. And if you can't do it a little bit faster, then you don't play with it. We're by no means experts at this. We can tell you our mistakes, what worked, what didn't work, what we think, maybe what's in the literature. But I think it's going to be important when you go back, you just start trying out different things. And QuickMD will help you be able to set up the system and get there a little bit faster. Okay, so now let me go back. End of sermon, begin of talk. Okay, so as I said, I'm Zan Luthey Schulten. I have a number of appointments, but the two most important ones are that I'm one of the co-PIs of our NIH center here, as well as the co-director of a National Science Foundation Center for the Physics of Living Cells. So, as I said, these reactions that set the genetic code, they're near and dear to me. I've been working on them perhaps too many years. First, I did it at a molecular level, and then of late have started to work on them in a cellular picture. So what are these protein RNA complexes? First, a little bit of a biology lesson. So it's these proteins that set the genetic code. Now, this is an amino-acyl tRNA synthetase. The amino acid that it's going to load onto this tRNA is going to be aspartic acid. And this tRNA carries the anticodon down here, those yellow yellow nucleotides there, they contain the anticodon, right? Now, I've color-coded it by using some of the techniques that are in multi-seq, which is in DMD. So I think I colored it by conservation of those structures or over all the bacterial fields, right? You can also color it by conservation of sequence, right? So these are the guys that are setting the genetic code. They have to read the anticodon and put the right amino acid here in the active site. And that's a long distance away. So the signaling, the communication networks are extremely important. Once you have charged the tRNA, it gets transported to the ribosome with this elongation factor, this EFTU. And here I've color-coded the interface with this. It's a GTPase domain there. I've colored it by conservation over all three domains of life. So this one poor little molecule, EFTU, has to recognize all the tRNAs, dock to them, and take them to the ribosome. Again, just a little bit for the biology, because it's in the picture. These space-filling nucleotides just show you where the modifications are on the rRNA. So even though we teach in the elementary classes there are 4 kinds of bases, there's 110 at least. And they differ between the various domains of life. So if you're really interested in understanding how these things function, you might want to take those base modifications into account. So as I said, where it goes next is now to the ribosome, that also got a Nobel Prize in chemistry several years ago, before they got the Nobel Prize for molecular dynamics. And everything's getting smaller. So these are like 3 tRNAs. They're reading a messenger that's actually on the small subunit, which I haven't even showed you here. And this is the whole large subunit of the ribosome, made up of rRNA and of proteins. Now, simulating the entire ribosome, that is a very difficult and long task. And so this is actually what drove me to start looking at cell simulations, because cells typically have, even the smallest one, have anywhere from 600 to 200,000 of these ribosomes in them. So that's another story. I will be having actually a workshop on the cell simulations at the end of the summer. But what I think is interesting is really studying things as they participate in the cell is a multi-scale problem, because there are many reactions that take place. We don't know the mechanisms. That's where the QMMM is going to come in handy. Some of them have been measured by the biochemists, but you really need to have an idea about the mechanisms. All right, so what I'm first going to explain to you is how we looked at the communication pathway classically. And then we'll make a quick statement about how you can do it quantum mechanically. And by doing that, it also sets up the regions near that active site, which you should probably include in your quantum mechanical calculation. In the case that we published in this Nature Methods paper, we didn't do that because that would entail a much larger calculation, and we wanted to get that through the review process as quickly as possible. So in principle, we should go back and even try to enlarge the QM region of what we've done. All right, so if you're interested in work that's on these molecules, this is the one that sets the genetic code. You screw it up there, there's no checking the rest of the way. In fact, people exploit that. Sometimes they'll try to put an unnatural amino acid onto a tRNA, hijack the anticodon for it, and introduce it into a growing protein chain. So there are a number of papers that we were very fortunate. On this campus, we had one of the leading evolutionary biologists, Carl Rose. We worked with him for several of these in the early 2000s. But the ones that are most applicable to the example that's in the Nature Methods paper are the capture and selection of ATP into the synthetase active sites, because to get the amino acid onto the tRNA, you have to charge it and make it become an adenylate. And then, of course, there's the NAMD goes quantum, which does a very detailed simulation, detailed QMMM analysis of the mechanisms. If you're going to do molecular dynamic simulations on rRNA and and with enzymes, you do have to think a bit about how you solvate them and the effects of the ions on them. Typically, the ions that are reported in the PDB structures are way beyond physiological concentrations. They put them there to help them crystallize the structure, because RNA is a bit more flexible than you're used to with protein structures. So on one side, you're going to see the simulation of a more physiological concentration, say of magnesium, and on the right, you'll see what would happen if you just try to use the ones that are typically in the crystallization process. So if you look at the tRNA and that stem loop leading to that anticodon region, it flips open every once in a while under physiological conditions, and it stays solid as a rock under these higher conditions. All right, so just a quick comment about how we did the classical simulations. These are in these early papers. There were crystal structures of the tRNA bound to, say, the GluRS with an adenylate analog in it. As I said, because it's important in setting the genetic code, there are many crystal structures that have become available. And actually, one of the colleagues who won the Nobel Prize for the ribosome, he was one of the first to actually look and publish a structure on this. So when we carried out those early papers, we were using the Charm 27 force field. You could also use amber. We were using the NTP ensemble with it. Later on in this particular picture, which I showed, we did not only the GluRS and with the tRNA, we also let the EFTU come up next to it and try to investigate the handoff of the charged tRNA to the next molecule. Okay. Of course, you put in the ions, you solvate it, and then you simulate as long as you can do. All right, so now let's zoom in a little bit more about just focus on these synthetases. And what goes on there, as I said, here's the tRNA, here's the anticodon arm, here's the anticodon, and it interacts with the so-called anticodon arm. Well, here's a simplified picture of this, but here's where your anticodon is, and then here's your acceptor stem, because it's going to be also accepting and putting on the amino acid at this end here. And the reason we looked first at these contacts with the protein is through really, literally decades of work on these molecules. They had identified particular identity elements that help the synthetase recognize I've got the right tRNA. In fact, when they tried to hijack the tRNAs to put an unnatural amino acid on, they had to go in and fool with many of these identity elements. Okay, so here's the overall charging reaction. Into the active site of this synthetase, you're going to have to bring an ATP, the amino acid, the tRNA, and then what should go out as a product is your charged tRNA, and you'll have an AMP and a pyrophosphate coming out there. Now, exactly how does it do it? There are roughly 20 of these proteins. There's one for each amino acid. The mechanisms differ a bit. There are in class 1s and class 2 synthetases, and the class 1 have a very similar active site topology in that, by that I mean in the active site, maybe one has a set of parallel beta strands and the other one is anti-parallel, but there are generally two classes. But then you have to ask the question, you know, there are a couple things coming in here. What has to be there for the reaction of the charging to take place? Well, with GluRS, you cannot charge, you can't even get the charging of the ATP or the glutamate with the ATP without the tRNA being present, right? The others, not so necessary. And we had looked at also just the binding of ATP into the active site of that. We used another free energy method. That's a Jack's paper. That was one of the ones that I listed before. Okay, so let's take a look how that goes. So here's the glutamic acid, here's the ATP, here's the tRNA, here's the synthetase. So now the tRNA will dock to it, and now the amino acid will come, and you'll have the ATP, and then now you're in the position to be able to charge the amino acid. You make it an adenylate, and you do that, and when you do that, the pyrophosphate comes out of the system. So now you have an active site with the adenylate there, and the tRNA, and what will happen next is, now you see you can go down here, if this is the 3' end of the tRNAs, and all tRNAs end in CCA, this is the A of that terminal group, then you've got a choice. Here's the adenylate, it either can add to this OH or to this OH, right? And in the synthetases, some do 2', some of them do the 3' OH. So we wanted to investigate that along with the protonation state of this amino group. So at the end of the day, we ended up looking at 4 different scenarios, and each of them we investigated using QMMM calculations, I think roughly 100 picoseconds, maybe a little bit more. But one of the questions we wanted to answer, because we had done this classically already, is how does the anticodon domain communicate with the active site? And this is where you can come in and use those 2 quantum sites and start looking at this. And I'll show you first how we did the network theory classically. We repeated it quantum mechanically. The answer is it's very similar, thank God. And there were a few differences, but essentially you can use the same protocol that we established to do the network analysis classically. You can use it with the quantum mechanical calculations. Of course, you won't be able to calculate quite as long as we did. All right, so how do you build this network? Well, a network needs nodes, and the nodes are going to typically be some atom on the protein and some atom on the tRNA. For purposes of demonstrations, I just took the C alpha of the protein and maybe the phosphate on the nucleotides. You really need to take 2 on the nucleotides, 1 maybe on the phosphate, at least 1 on the base. And we explained that in there was a PNAS paper about doing this network analysis. So those will be the nodes. Well, what about what connects them? Well, you look at your MD or your QMM calculation, and you see how many of these nodes are in contact over a certain percentage of your simulation. And by I say contact, what I meant is like any atom on that on a residue where you're looking at the C alpha with any atom on that nucleotide. Are they in contact or make contact over, say, 75% of the simulation? If they are, draw a line. That's going to be the edges of your network. So you got nodes and you have edges. Right now, I've drawn them all of equal weights, right? But what you want to do is use the information that you have from your molecular dynamics simulations. And what you have is the information about correlations. The fact that something moves together or anti-correlated or correlated, that's information, right? So what we're going to do is we're going to go weight those edges by using the correlations from your simulations, okay? Well, not yet. First, we're going to identify maybe some interesting points in this network. So, like, if we keep with the synthetase example, like one of these could say be the maybe the anticodon domain, and this one could be a residue on that active site, right? So, and this is a path that would take you 10 steps to get there. But I think you can also see there are other paths. This might take a little longer. There's some that are longer. Or you could go like this. This would be much longer, right? So the length of that path is also going to be important, particularly, as I said, when you now get to the point where you weight them. So first, let's get a look at a correlation map for this thing. That's on the right-hand side. And the first part of it is the atoms in the tRNA, and the rest of it are the atoms in the protein. So you're looking at these correlations. This was done with Pearson correlations. You can get better results, less noise in them if you'll use generalized correlations. We don't have that software out yet, but Marcelo has adapted it from what we did in there's a tutorial for doing this, but as I said, you get better results when you use the generalized correlations on them. So now let's go take that information and start coloring our network. And first, this would be without weighting. Those were the contacts that are made 75% of the time over the trajectory. And then if you weight them, this is the picture you get. And I think it's interesting. You can already start seeing this, where the tRNA interacts with the anticodon domain is very important. And also there seems to be some clustering going here. In fact, you have over various regions communities coming in. And now I have to go over and use the language of network theory, because the network analysis tool that we borrowed here came from an article in PNAS. It was used to study social networks, like are you a member of the Boy Scouts, the Republican Party, and whatever features that you want to do. So it's Gurman-Newman, as it's called, the two authors who developed it. Right? And what happens when you have the communities, you have more contacts within a community than between the communities. That's the simplest definition of these communities. All right, and there's the correlation function that we used. All right, so now let's just look at some of those shortest paths, say between maybe that mid anticodon. Again, you may or might not know anything about setting the genetic code, but there are 3 nucleotides that have to be read. They are in positions 34, 35, 36, and let's say look at the center one and its connections, and connect all the paths from there up to the active site, right? And you'll see that there are many paths that look rather, that are short, but one of them is going to be the shortest, and you'll call that the optimal path, and we'll rotate it around. Right? And then the others are interesting, and when we did in the original PNAS paper where we published this, we did some mutation studies, so then you could try to predict what influence the mutation might have on the binding. But I will tell you there needs to be a theory of these network analysis that associates it completely well to getting us a KD out of this calculation. All right, so you have that information. Again, as I said, this is just to tell you again what these communities are. This makes is a very simple example. We have like 3 communities, and there looks like a single link connecting them. And in fact, what the Gurman-Newman algorithm does, it goes about then cutting bonds and sees if it falls into independent communities. So that's how you find them. And I won't bother you with the rest of the analysis. I'll just zoom in now to the active site. But we could also analyze the whole communication within the proteins and the RNA, because at that time we were working with Carl Rose, and we were interested in how maybe this communication evolved. So that was a question we were trying to answer also. All right. So now let's go over and look at this mechanism. As I said, it was initially suggested by Tom Steitz, who would go on to win the Nobel Prize for the ribosome. So we're back to the A76 on the tRNA, the end of it. We now have the adenylate in here, so you'll see there's the AMP moiety over here, and then there's the amino acid moiety over here. So this, the mechanism that he had suggested is that when this thing splits apart, you're having this oxygen here is now bound to the OH group there, and that hydrogen has come down and now is on the AMP moiety, and then this should be leaving the active site. Turns out that it's leaving, even classically to calculate this, took over 100 nanoseconds, and we never got it out of the active site completely. And that is a large part of the energetics of understanding this reaction. We did see after about 100, 200 nanoseconds, you could see the tRNA beginning to peel off the protein, but that is a very lengthy process getting rid of this group and getting it solvated and getting it out of the protein. All right, so we did the community analysis on this system, and then we just want to speak now about the one that's in the active site. So let's zoom in on that. And you'll see that there are two major communities. One is purple and one is gold. And if you look at what they are interacting with, one part is interacting with the AMP moiety, and one part is going to interact with the amino acid moiety. So there you see, that's the part interacting with the amino acid, and then this will be the part that's interacting with the AMP. This, to me, suggested automatically what we should perhaps be using for the quantum regions, these two networks. But as I said, rather large, and when one did the simulations, whoops, I think it's the next slide, I think we only did it like for 236 atoms. This would have been a bit more, let's put it that way. And to be frank, we might have even gone back and taken a more intense look, like how many of these nearest neighbors could we have done? Do we really need all these others that are down here? Okay, so now that's all classical. We have an idea about the communities, what we should include. Now let's go think quantum mechanically. So as Marcelo nicely explained, you're going to have to decide on a QM region and an MM region. Then you're going to have to decide how those two guys are going to communicate with one another. He went over the various ways we have of communicating with them, redistributing the charges over here. I won't repeat this because he did, as I said, he did a very nice job of explaining this link atom notion and these ways to redistribute the charges over on the mechanical part. So I think we offer 1, 2, 3, 4, 5 different methods there. This is the one that was chosen in the paper for the simulation on the synthetase. And as I said, we investigated then these various scenarios, having it the amino acid be added to the 2' or the 3' group. The quantum mechanical part was 236 atoms. We used the PM7. So I hope that, Garrett, you'll explain what PM7 is tomorrow. Thank you. As I said, I did about 100 picoseconds simulations on those. Now, great. Why do you do this? You just want to jiggle it? No, we would like to get a free energy for that reaction. So, how are you going to do that? Well, we got a couple different mechanisms. You could just put everything there and just wait. Does something happen? And hope something happens. Waiting's not a good idea. Particularly because most of these reactions, unless they have really small barriers, they're not going to take place immediately. So you got to select a mechanism, right? And you want to probe that mechanism. So that automatically says to me, if you're going to probe a mechanism, but you've got to decide on what distances are important. So if many of you run free energy calculations? Who hasn't run them yet? Right? Okay, so when you do these, I don't care if you use this adaptive biasing forces that we have. I myself am a little bit more partial to metadynamics, but you've got a problem. You've got to pick reaction coordinates. You have to pick, and there's just normally not one distance, so you're doing a couple, so that's why they call it collective variables. So we've got to go pick, well, do I just look at that distance, or that distance, or that distance? What do I include here? Right? So that's the next thing you have to do is probe it. And then, so when you draw these pictures, when they're trying to get at the free energy, they put reaction coordinate here. That's why they never specify. It's just normally never some simple distance. It's a whole bunch of things that you're looking at. And I think this is something, hopefully, Rafi, you're going to explain in detail. So I'll just run. So imagine you had, like in this case, 7 different distances. Where you can look at the distance, say, between this oxygen and this carbon, or that hydrogen getting over here. So 7 different distances. And guess what? Some of them will be more interesting for one mechanism than they will be for the other mechanism. So one of the things you might ask, well, hey, here's one of those guys. It doesn't change at all over the course of the reaction. Why would you choose that? You think a good reaction coordinate ought to be one that changes. You know, like I'm in state A and I go to state B, boom, a big difference in that, right? The trouble is, if we're going to check a couple of different scenarios, you've got to use the same set of collective variables, right? And as I said, it's going to be, this might be boring for this 2' addition, but hey, look, you get change when you go over and do the 3'. And in the same way, the orange, that's not too bad, actually. The green is a little boring here, much more interesting over here. So you've got to, once you decide on a bunch of collective variables, and I'm telling you, this part is, nobody can tell you what to do there. In fact, if you've ever done metadynamics or well-tempered metadynamics, it's choosing the right reaction coordinate is half the battle for these free energy methods. And there's where your intuition has to come into account. That's why I say the fact that if you could do these calculations a little bit faster, then you could probe more things. Right? Or, as I said, once you decide and you look at the free energy, maybe which one of these is the scenarios is the best one to do, then maybe go back and do even a more intensive study of it. All right. So what you're interested in is how these coordinates change over the course of the reaction. Again, Rafi will discuss this. So if you guide it along these mechanisms and then carry out the optimizations and do the free energy analysis, you'll get these free energy curves here, and you'll see they'll have different profiles to them. This one looks pretty high. That goes up to almost 35. This is like around 25. This looks like it's under 20. That's going to be the one with the lowest barrier, right? So you start calculating, but you look, this endpoint is still not below where you started, and it won't be until you allow that HAMP to get solvated and move further out of the active site, right? But this was taking way too long to keep on doing that with the full quantum mechanical, molecular mechanical, the QMMM method. So we switched over then here to doing then just the classical mechanical approach to doing this problem, right? And then that's what you're seeing here. And as I said, I think in almost all these reactions that are involving hydrolysis of ATP or GTP, you're going to find out that the rate determining step is getting that stupid thing, the phosphate, hydrated and out of the active site. Right? Okay, so I'm sure you're going to be shown this movie several times, but I love it for this reaction. And I think more than once Marcelo and Rafi cursed me for picking this reaction. But it makes such a great movie. So you'll see the bonds, so you'll see the hydrogen moving and then the amino acid being attached. And Rafi did just an incredible job of getting this movie. So there's our system. And now let's zoom into the active site. Okay, let's rotate it for a second. Pedagogically, it's fantastic. So you see the AMP moiety over here. They'll even label it for you. See the dotted lines indicate where the action is going to be. AMP, there's the amino acid, and there's the sugar at the 3' OH on that A76. And now let's watch the reaction take place. And you're not going to want to put up all the orbitals. You know, you're going to be selective. And I don't know if Till you showed them QuickMD, how you can select whether you do HOMOs or LUMOs or which ones you do. But now, any moment now, that hydrogen is going to go over there. The suspense is really building here. Bang! Okay, great. And now we have to then connect the amino acid to the tRNA. That's coming next. So this looks like it is a two-step. So you formed the bond, and now the bond to the AMP moiety has been broken. Right? So there you are. That's a fantastic representation. And I do hope for those of you who teach will also use this in your classes. All right, and then it just remains for me again to thank all the people who contributed to getting this work out. As I said, everybody's role was important. You know, being able to watch the reactions, being able to see them visually, I think is an extremely important part. Having a MOPAC available to us so that it works so quickly is an incredible gift. And so I hope you guys will make good use of it. Rafi and Marcelo set up a very nice tutorial. You're not going to do this problem, it's too complicated for the workshop. Go do something easier but equally important and go through the steps. And I think, just as Marcelo was saying, it's important when you're using QuickMD that you go through and look at each of those steps. What are the charges? I even taught this in my class this semester, my computational biophysics. And the students, they had a little bit of trouble. Our tutorials weren't so good then. But I think it is completely doable in the time that you guys will have here. And as I said, you've got a room full of experts here to help you, so you'll be in really good shape. Okay, so are there any questions on the system? No? Everything crystal clear. Okay, you're really looking forward to working on these things, right? So I assume you all came with your own systems. Okay.
Can you just remind us what the how the network theory integrated into the simulation was there?
Okay. We had from the classical mechanics the network theory that said that there should be a group of residues, or atoms either on the nucleotide, nucleic acid, or the protein that are interacting with that adenylate moiety. That wasn't. So first we wanted to check that in the QMMM calculations, and it did. The region's very similar, but as I said, ideally, if I could have done this over again, I would have tried to even use the network theory to decide which of the surrounding atoms I would include in the quantum part.
I think it's very powerful.
Yeah. Given the provision, you've got to look and see like, oh, you can't cut it somewhere that I have -2 in my quantum region here, right? So, but use it to guide you. And I think the network theory is extremely powerful technique. And it just, you can't. Use it to guide. Is that person agreeing with me? Yeah. So as I said, I wish.
Just reminding you that it's recorded, and I can go back and just watch your lecture again
Yeah.
instead of
So
asking you to
I
repeat
will
yourself,
warn
which I
you.
apologize for.
We had the network tutorial as it is. I have not updated it in many years because I had to wait till I had such a good student like Marcelo, and I would give this talk. The last time I gave it was in Germany, and this Max Planck director, who was a former student of my husband, said, Sam, you should really do generalized correlations. So it took us a while to get that written and implemented into the program. And to be frank, we might even replace I did it on the synthetase. We'll probably put in the QMM correlation map. It's in the Nature Methods paper if you want to look at it.
Right. But the network theory is exact. Is that correct? There's no approximation baked into it as there is.
Well, if you use the Gurman-Newman,
yeah.
it is. There are no adjustable parameters. The thing is, you do not want to use it on the whole ribosome. Tried that, right? There's just the network is so large. So I looked at there was a French group that came up with a network analysis program. That they use it to look at cell phone usage, right? But it's there are a few adjustable parameters in it, and I tried to see if I could adjust them to make them give me the same Girl and Newman results. Couldn't, so I sort of lost a little interest to it. But there are other methods to handle really big systems. So if you're somebody who really wants to put this in a, protein, active sites in a protein embedded in a membrane or do the ribosome where the networks get really big, one might want to look at one of these other methods to get the network. But it's most important to get the communities near the active site.